Well, I want to thank everyone who went before me because this made my job easier. A lot of you set the stage for what I'm going to talk about. And I'm the last talk before lunch, which is always a weird spot to be in. So I promise there's lots of videos of kids, some kids you might recognize. Um, so I wanted to just kind of start by kind of showcasing where I came from. So I'm currently at the University of South Carolina. But I have a very different background than I think many people in the room. There's a couple of you that might be a little bit more similar to me. Um, I'm what we call a developmental cognitive neuroscientist, which basically means that I'm obsessed with learning about how the brain is developing and changing, right? So we look at how brain function emerges across all of these different life stages. So we're really interested not only in like what you're seeing now, but how that's going to change, and particularly trying to understand how if we can kind of capture when we have elevated risk for some sort of shift in neurocognitive abilities, that we can think about that and intervene in a way to put our kids back on a neurotypical track. So when we think through kind of, and everyone else has kind of done a great job of like walking through this clinical trials lifespan, so I won't belabor this, but I see myself kind of sitting in between these two pieces where we're really kind of thinking about these early, like low level basic mechanisms, but understanding how we see that in the human brain. So kind of taking all of this great work that we're seeing from the cells and seeing from our animals, trying to understand how that then represents what we're seeing in the brains of our kiddos. So today I'm going to walk through one of our studies that we've pretty much completed, and hopefully sell you on this idea that EEG can be a really great biomarker, although now I have some ideas about hemoglobin and near-infrared spectroscopy, so we'll have to talk later. And I really want to showcase one of the ways that we sought out to kind of reduce the burden to participating in research. I think this was mentioned earlier, that it is a lot. We are asking a lot of our parents. So we've adopted a really weird way of doing this. So I'm going to showcase what we did in the States and how we hope to kind of continue this work. And then hopefully, if I have enough time, I meant to time myself, I will share some brand new findings that I have that really do link this cross-species piece, working with Annette Schenk in the Netherlands. So we've already kind of talked through this. Everyone has a different way of displaying how we think about the body and how we measure different things in the body. I go vertical. So you see kind of this idea that we're largely, I'm coming at this from an autism perspective, just from my training. That's where I kind of started in this world. And so we know that that's diagnosed with behavior, but we can use the brain to measure more discrete, more nuanced pieces. And then the idea is that that's going to link down to things like protein expression, protein-protein interactions, and all of the things down to kind of our genotype. So when we think about making brain biomarkers, for instance, thinking about autism, the idea is really to say, okay, so we want to find a marker that says this is autism, this is not neurotypical development, right? So that's kind of the premise of what we're working with. We use lots of big terms trying to talk about like the validity of this, that this isn't just something you measured once, that we see this over and over again, that it's a stable biomarker. And we're not doing this from scratch. We're really building off of some pretty big autistic consortiums that are really meant to track neural biomarkers using EEG like I do, as well as eye tracking, which we actually do in the lab as well. But there's some problems with this, right? So a lot of these large scale consortiums are not even seeing our kids, right? So first of all, a lot of kids, there's questions about whether or not if you have a GRIN diagnosis, should you be included in these idiopathic autism studies, right? So this is kind of a new question out there. But in addition, our kids tend to have more challenging behaviors. They tend to have communication difficulties, more sensory sensitivity, so they might not necessarily like tolerating the net. And a lot of this is like putting kids in front of a computer screen and having them press buttons. I don't know if you parents are thinking of your child, if they would sit there and press a button box. That's not going to work for us. And the other piece that I brought this up last year at our GRIN 2B conference in Chicago. I've been kind of coming at this from the perspective of, well, how is there a GRIN 2B autism biomarker? And that's probably not exactly what we want to be doing, right? So we really want to be honing in on what is going to be specifically, and it might have some links and ties and relate back to our autistic biomarkers, but they might not necessarily be one and the same. So the way that we kind of conceptualize how we work in this space is we think about GRIN2B, which, and I should have mentioned, I'm very interested in all the GRINs, but this is just the area where I've had funding and support. So we're growing, so don't be disheartened. But we think about GRIN2B, we can think about how all of these different pieces kind of lead us back up to what we see in our kids. We see learning and language delays. We see autistic traits, if not an autism diagnosis. There's lots of hypotonia, motor delay, lots of cerebral palsy diagnoses, microcephaly, right? So that's kind of where our doctors are telling us with their kind of validated measures. And there's actually a little bit of work that's really kind of looking at the brain structures themselves. So there are some abnormal structural differences, particularly in the amygdala, which is this little arrow is pointing to the amygdala here. I'm less familiar with the zebrafish. I'm excited to learn more tomorrow, but we do see that the same kind of features we're seeing this common thread that the brain is an important place to be. So some of the work that we did in my lab, and we recently just resubmitted this paper, is we asked parents about their kids and we filled out the standard measures, many of you have maybe done before, to look at what they're reporting as their child's strengths and weaknesses. And what we see in GRIN2B is actually fewer social problems. So this is really kind of re-emphasizing the fact that maybe this autism diagnostic kind of track is not appropriate for GRIN2B, because we see these beautiful strengths in social and emotional exchange. And when we actually look into what our kids are doing, so not only just looking at the parent reports, we went back and reviewed all of our clinical reports and pulled out what the strengths that we talked about in the kiddos that we saw. And we really kind of saw this reiteration of high social motivation. These are individuals, these kids want to be with other people, but they might not necessarily know how to do that appropriately, right? So there's that where that autistic symptomology comes in. We see unusual nonverbal communication, so they're not necessarily using nonverbal cues in the right way, and they tend to be sensory seeking. So this is just a representation. So this is from the social responsiveness scale, and here higher scores are indicative of problems. And so you can see GRIN2B sits at that kind of top line here, which is kind of our borderline. It's actually our clinical line here, and the dashed line is our borderline area. But if we dig into the social motivation scale, you see how much lower the GRIN2B group is relative to our other groups. So there's really clearly something here related to social motivation. So I'm excited to tell you something that we put together yesterday, which is that my brand new postdoc, Dr. Kaylee Nelson, has just submitted her first grant in partnership with the GRIN2B Foundation back in the States and Uplifting Athletes to look specifically at social motivation. So if you're a parent in the room, I'll put this slide back up again at the very end if you want. We actually have an opportunity for you to participate. We're opening this to all GRINs, so we'll come back to this, and I'm happy to kind of chat with anyone about this study. But we're really trying to understand what are those, from a strengths-based approach, what are our kids looking like? Why are they doing this amazing thing, and how are they connecting, and what does that look like across different ages and across different GRIN disorders? All right, so back to what I meant to talk about today is thinking about how do we connect across all these pieces. So as a brain scientist, I recognize the fact that there's kind of this gap here in how the brain is functionally processing in real time, right, in our human kiddos. So what I'm trying to do is bridge between our genotype and our phenotype by narrowing in on an area that we think is going to be really helpful, not only for understanding the brain in our kiddos, but linking back to our animal models, our preclinical models in a way that we can actually make some sense and then use those preclinical models in a way that is going to truly help accelerate intervention and treatment. So the study that I designed, I call it the BioGene study, and the website's here. There's an actual GRIN2B page, and now that I'm part of the larger GRIN community, we'll probably be opening that up a little bit more broadly, but I didn't fix that in time for today. But I'm building off of work that I did in my postdoc at the University of Washington. So if any of you participated in the tiger study, have heard about the tiger study, this is really what we're starting from, right? So it's that same kind of idea of understanding deep phenotyping, and I'm just adding in that brain piece. So we're really trying to do deep brain phenotyping. So what is EEG? For those of you who have never maybe seen what this is not necessarily what you would do for an epilepsy screener. We use these high density nets that go on very, very quickly. I started this work with babies, so I'm really used to nonverbal, preverbal kids. And so we have a system that goes on as quickly as possible because we're not going to glue anything. Our kids aren't going to sit that long, right? So what EEG actually does, for anyone who's curious, if you have a part of your brain that is specifically doing some work and you're having this thought, so this is kind of supplementary motor cortex, what happens is that patch of neurons in your brain send out electric signals. And so what we're doing with the EEG is we put a net on top of your scalp, and then we're just recording those electric signals across the scalp, okay? So what's kind of weird, and this is where, like, if you want to geek out with me about EEG later, I'm happy to. The reason why we love the high density part is because how that electricity shifts in real time at the microsecond level is really important. So that same piece of brain activation might send signals to this lower part in a negative way, but it might send positive signals in the other direction. So we have a complex math problem on our hand, but thankfully we have really cool computers to help with that. So what we did is, because pandemic hit, I knew that people weren't going to be able to travel to me, is that I packed up my car in the fall of 2021 and I drove around the United States. So in a matter of 92 days, I traveled over 15,000 miles, and I meant to convert this to kilometers and I forgot, but it's a lot, okay? And we ended up seeing 55 probands that target kids, and we had one parent who also had the event. So that parent's two children had an inherited event. So you can see we're busy. We made some loops because I had to go home to see my family at some point. But it was a tremendous success. And we're not necessarily done. I did just kind of finish relocating to a new institution. So now we've got some hearts of the places we kind of want to go next. So we do plan to be in Denver for the GRIN 2B meeting. And we're really excited to come back to Europe. I didn't bring my EEG with me this time, but that's just cause to come back next time. So of the 55 kids that we saw, I just want to emphasize that a lot of these kids were non-ambulatory, were either in wheelchairs or were otherwise really kind of restricted motor-wise, which parents know that makes traveling anywhere just that much more of a kind of concern. And we actually had a dominant group of that were nonverbal. So a lot of these kids were not necessarily using verbal speech, but almost every single kid made it through at least one of our experiments. And our experiments are between 5 to 25 minutes if they really want to sit through all of it. But if we can get 10 minutes of data, that's really, really great for us. So from this road trip, 27 of these kids had a GRIN 2B mutation. I was also collecting data for SCN2A at the same time. And so this is just a little kind of snapshot of what this was like. So this is me practicing putting together the system, because I would bring it into people's houses and have to set it up. We would look through the house, kind of find the right spot where the kid was going to feel great. So you'll see we're here, we're in the kitchen, we're on the couch, we're in kind of like the work chair in the kitchen. And showcasing how quickly these nets can go on, and for a variety of different skill levels and attention levels. We had a lot of fun. We had some challenges. Not everyone was as excited to see me as I was to see them, which is okay. I wasn't offended. But for the most part, all the families seemed to really enjoy this kind of participation. We had some kids fall asleep, which is also okay for what we were doing. The rest of this video is on the website. I just want to highlight a couple of things. So one of the things that was really clear right away is that we have to adapt to what each kiddo needs. So this kiddo on the left, her happy time is between 6 and 6:30 in the morning. Now, I'm a night owl, so that is not my happy time, but I was at her house at 5 o'clock in the morning to set up our equipment. So when she woke up, we had pancake breakfast together. And for the time it took her to eat 3 pancakes, we did all that we needed to do. She was total trooper. We had another kiddo who really just was uncomfortable with it. And we had sent home like a practice net to kind of get her used to the situation. And then I realized she just wanted to see somebody doing it. So I put the net on myself while it was way too small for my head. So I ran the whole session with a net on my head, and we made it through. She took a little bit more time because we took more breaks, but she really had a great time. And then this is a kiddo I've seen multiple times before. She's done this with me before, and she was excited to see me. That's social motivation, right? But then I put the net on and showed it to her because she was so excited previously. She was not as happy. But I'm happy to report that she loved it. By the end of the session, she was full smiles, and I took the net off while mom was still doing surveys and I was starting to pack up. She wanted me to put the net back on. So we played a game where every like 5 minutes I was putting the net back on. So the kids actually get a kick out of this, I think, to a certain extent. So what biomarkers did we choose to target? And we had a lot of different things we could pick, but we really kind of leaned into this idea of wanting to learn a little bit more about language and learning, and so how ongoing processing is happening. And we have some evidence and reasons to do that. So we decided to kind of focus in on some really, really basic biomarkers as a first start. So we're looking at attention signals, which are also very, very easy and appropriate to measure in our animal model. So we have this nice translational opportunity. This is kind of what our tasks look like. So here's Natalie listening to the sounds. They're quiet, but basically you have one high sound that's repeated and then a low sound that's what we call a deviant. And what I want you to pay attention to is there's these like novel chirps and squeaks and organ noises. We call those novel sounds. And we also did a similar experiment. For time's sake, I won't go through this, but we were also looking at speech discrimination as well. So what we do with our EEG signals is every time that you hear one of those sounds, we say, let's look at that EEG pattern for the next half a second, 500 milliseconds. I don't even know that I could like actually understand how fast this is. But what's really kind of cool is that if we time lock to these auditory signals, we actually not only get one, but multiple different ways of looking at how our brain is processing this information. So there's this first kind of positive bump here that doesn't look like much to you probably, but this is an attention signal. We have some early detection signals down here, what we call the N1 component. But what I'm going to focus on today is what I would consider our attention orienting response. So particularly when you hear that thing that pops out of nowhere, that like chime and squelch, your attention kind of like gloms onto it, right? And so we see this kind of big shift, and it's great because it usually is a very big effect. So even if we have a kiddo who has more variability from child to child, we can still capture this effect. So largely what we're trying to see and predict is that we expect that the novel sounds are going to have a higher amplitude, so kind of have more voltage than the frequent tone, which is kind of down here in this picture. And so what we found in GRIN 2B is actually ended up being more of a mixed bag as I started to add in some of our new families to this. So this includes our road trip GRIN 2B folks, as well as about 10 other kiddos and individuals that we saw over the past couple of years. And what you can see, like you see how the autism, this is an idiopathic autism group and a neurotypical group. You see how the red is truly higher. I should have flipped these around, but a lot of these kids with GRIN 2B, each line is one kid, aren't showing differences, although we do see some, right? So this kiddo right here in particular has a very big, and I would say an autistic-like pattern that we're seeing. So we're not actually seeing, and that's just from the tones experiments. So we're not necessarily seeing a specific condition effect here. We're just seeing that it's not like the neurotypical group and not like the autistic comparison group, but this is consistent with some of that work that we saw, talking about the hippocampus, issues with memory and learning. And because of the fact that attention is at the basis of all of these things, if your attention system is not acting the way we expect it to, then all the other ways in which you're going to kind of process information are going to be affected. So that's what I mean by low-level skill, is that it sits at the bottom. You need to be able to attend to something if you're then going to be able to understand and start to process language or a math problem or the different processes to brushing your hair, right? So in I think I have 5 minutes maybe, I'm going to try to kind of talk a little bit about our next steps, because I think that what we need to do is dig more closely into this. So we are going to be adding some more GRIN to be participants as we go along. We are going to be adding some controls, because we actually don't have the controls for the speech test, which is why I'm not focusing on it today. But I've been working with colleagues in the Netherlands, Annette Schenk and Michaela Finkova. And Laura Block is an advanced graduate student. So she's just about done with her dissertation, which is the work that she's given me permission to present here. And what's really great is, and they let me at some point sex Drosophila, which was really fun as a human scientist. So what Laura is really looking at is kind of understanding not only the attention signals, but how does that attention signal change over time, right? So it makes sense that we would want to attend to something right away and have a big response, but neurotypical brains start to filter that information out because it's no longer as efficient or as important. So we have this big initial pop out, and then it kind of filters down. And they can measure this with the fruit flies just as well as we can measure this with the humans. So what Laura did is she took a series of different knockdown flies, and she actually was modeling this across a lot of different neurodevelopmental genes, many of which are going to be in Simon Searchlight. Here she is, and she takes each fly, and she has this great process where she puts them in these little tubes. And then what she does is she flashes a light. We turn off all the other lights, and she flashes a light. And she looks to see how much they're jumping when they have this aversive light flash. And what you kind of end up, there's a smooth bit, but I even smooth it even more, is that there's the blue line here is talking about unaffected flies that are kind of jumping at what I would call like a neurotypical rate. There's some flies that either don't jump at all or have this really, really flat pattern where they hardly jump. But what we're seeing is that there's what we're calling habituation deficit, where I also call it sticky attention, right? So you should be kind of filtering out this information, but you're stuck on this information, so it kind of remains at the forefront. So this is the actual data for GRIN here, so the NMDR2. So you can see the number of flies she has, and what she's measuring is like how many trials does it take before you start to habituate. And so we have a much later habituation period for the flies. And so what I did is I started to look at what are we seeing in our human kiddos. And what we actually see in GRIN2B, so this is taking the red is that big P3 response that's supposed to be bigger than the black response, that new sound. And actually, statistically across these 34 kids is that it's just a flat line. So there's no indication of habituation. So there's a habituation absence. And this is relative to what we see in ASD, which is more of that sticky attention where they do habituate, but it kind of is stickier. And in neurotypical development, where we see this kind of really early, rapid habituation. So there's a lot of things that we need to do from here, in part, like really starting to tease apart those individual differences. So that's literally what I'm doing this upcoming week. I was hoping to have it for you today, just didn't quite get it together. But we really want to understand, so what does it mean? So for each kid, we can pull out and tell you what their habituation pattern is. And so what does that actually relate to how much sleep they're getting, to how they perform in other cognitive tasks. And the Vineland seems to be a really, really important piece here. So we're kind of trying to map this back onto other adaptive skills from the Vineland, as well as sensory behaviors. But the other big problem here, and I think that tomorrow I'll have a lot of questions from everyone doing clinical trials, is how do we pick and how do we understand the developmental trajectories and the time course? Because the brain is changing in different ways, particularly when puberty hits. what's really happening there? It's really difficult to kind of know and anticipate, right? So what we want to be able to do is have a prognostic way of saying, okay, so with puberty, here's what we expect to see. And understanding how those kind of biological features of a person, like sex and gender, are also going to kind of play into how the brain is forming. So what I really want to do, so this is me plotting all of the kids that I saw in the past couple of years. So this is their first time point that we came in and we saw them. This is, so each row is just a different person across age. And what we ultimately want to do is create some sort of way to do a natural history study of the brain. So we're not necessarily just saying, here's the brain at one point, but here's the brain, how does it change to tomorrow, right? So what's the stability from tests from day to day, but then also how is that shifting 6 months down the way? And what I would be really excited about is, because we have this beautiful data, is we really would start to understand and have this opportunity to flesh out developmental trajectories, because we would have multiple time points overlap across kids as well. All right, so I'm just going to end. I don't think I have time to kind of go through all of these, but I'm looking forward to spending the next next day and a half to really kind of think through these pieces. Importantly, as somebody who hasn't been involved in the other GRINs yet, how can we get involved? What is it that we should be looking at? Is language and attention not the right phenotype? We also want to be thinking about, are there other pieces? So now I'm really excited about the blood, which I didn't know I would be. So maybe we need to be kind of thinking through that and understanding either the brain functionally through something like NIRS, and really being working together with partners. I'm glad to see a lot of folks that are on the clinical side here too, to think through how these different biomarkers that we heard in this kind of session, how they map onto what our kids can do. So I'm going to go ahead and end there, and I'm going to leave this slide up just for a little bit, if that's okay, while we kind of queue up for questions, just in case anybody wants to participate in the survey. And I'm happy to talk about what that part of the study would look like later. So thank you.